So for the last 30 years, we've told everyone that mites prefer nurse bees. This is something that you've probably heard in your beginning beekeeper classes, and it's what all research has told us. My research overturns about 30 years of honeybee Varroa research because we found out this is absolutely not true. All right, thanks, James. So as I got the introduction, so I'm a postdoc and I work with Jay Evans. We have a pretty dynamic lab here. So I'm in the bee research laboratory in Beltsville, and I work in a genomics lab, but I mostly study behavior, either behavior of the host or behavior of the parasite, and in this case, it's Varroa destructor. And we look at which behaviors are driving disease transmission. And so it's just a different way to look at how mites are killing your hives. And so we essentially approach the problem like a behavioral ecologist. But we're going to do this with a little introduction into Varroa. If you're a completely new beekeeper, then you'll get some information out of it. Otherwise, the first couple slides will be a recap for everyone. So pictured on the left, that is Varroa destructor adult female mite. That is the thing that we talk about more than we actually talk about bees themselves. Now, this is an ectoparasite, which means that it's an obligate parasite that has to be on a host to survive, and it lives outside the host. You can visibly see two mites here, which are feeding on an adult worker bee. And then over on the right, this is actually the bee that we tend to focus most on, excuse me, when we talk about Varroa destructor. When mites feed on adult bees, they are capable of transmitting viruses. They do this through their feeding. When they feed on developing brood, if a deformed wing virus, which gets its name from the crippled wings that it can cause in bees that emerge, if the virus migrates to the bee's brain, that bee can emerge from its pupal cell with deformed wings. And we really, as beekeepers, tend to focus on this bee quite a bit. They're very visible in our colonies and they stand out. They stand out for several reasons, not just because of their physical deformity, but also because they physically move different than every other bee in the colony. They're going to be extremely short-lived, though, very conversely compared to like this bee over here in the center, where in our experiments, we've seen that these bees can actually get bit as adults, show no symptoms whatsoever, and live for a long period of time. And in that conversation right there, I'm bringing up two really important terms, which is you can have symptomatic and asymptomatic carriers. So in any infectious disease system, when you have hosts, some of your hosts are going to be symptomatic. So that would be equivalent to someone with a runny nose and a cough showing up to work. You know they have the flu. But you can also have an asymptomatic carrier. So this would be the equivalent to someone like me who shows up infectious, but I'm not exhibiting any symptoms. And unfortunate thing in most infectious disease systems, the asymptomatic carrier is actually capable of getting a lot of other people sick. And we're going to talk about that later on. So Varroa reproduce in brood cells. The mites will enter what's called an L5 larval cell just before that cell caps. They predominantly prefer drone cells over worker cells. But they will invade worker cells, especially when drone cells become unavailable later in the season. When they enter that brood cell, they'll go underneath the larva and submerge themselves upside down in the brood food and hide. Once that cell gets capped, that larva is going to consume the rest of the brood food and turn itself over, and in a few hours start to spin. That releases the mite. The mite here in the center was actually excavated from a larval cell, and all I did was take a pair of dull tweezers and scrape away a capping and grab that fat larvae out of it that had already consumed the brood food. And when you take them out at that stage right there, at that L5 stage before they start spinning, they're always on that larva, so they're really easy to find. Now, we tend to notice invasion into brood cells when our bees engage in uncapping behavior. So pictured over on the right, these are pupal cells that have a mite inside those cells, and the bees are uncapping those cells because they can detect a mite. Now, we have bred our bees to do this because if we can interrupt mite reproduction, mites are entering these cells to reproduce, then we can suppress their ability to increase in numbers in our colonies. And so we call this suppressed mite reproduction or VSH behavior, and we largely view it as a good thing. I will point out that our group is doing work looking at the consequences of this behavior. And so we published in scientific reports that the cannibalization of infectious pupae by adult bees can cause a super spreader event amongst adult bees. And during my thesis, I also showed that when these adult bees uncap and then cannibalize that pupae, they can become infectious with the virus that pupae is harboring. When a mite feeds on that adult bee, she can acquire that virus and then transmit it to another pupae. So we actually showed in the laboratory that your mites can become more infectious when your adult bees cannibalize infectious pupae. So we're not saying, to stop keeping VSH bees. They've definitely shown their utility in our beekeeping operations, but we're actually showing some of the consequences of these behaviors as they occur in our honeybee colonies. We never want to look at a behavior when there's an infectious agent, in this case, infectious pupae, and think that there's no consequence of that pupae existing in the colony. I was really interested during my thesis of how viruses can be transmitted mite to mite. And so I read a lot of tick papers. And in ticks, pathogens can be transmitted from a naive to an infectious tick when they feed on, or from an infectious tick to a naive tick when they feed on the same host. Now, if you ever hear of a naive tick or a naive mite or a naive mosquito, it doesn't mean that it's silly or stupid. What it actually means is it doesn't have a pathogen inside of it. But those naive vectors, and a vector is any biting arthropod, so I just mentioned them, ticks, mites, mosquitoes, lice, when they feed on a host or they feed with another vector, they can acquire a pathogen. And so this is a really important thing in disease ecology. So in the laboratory, what I did was I hand-painted mites. So I have my orange mites. These are my control mites. They have no additional virus in them. And then I hand-painted some mites blue, and we had them feed on a pupae that was injected with a genetically modified form of deformed wing virus. And so I have essentially a color-marked virus. It glows blue. And we have color-marked mites. And what I did was I inserted them into larval cells in the laboratory and then recapped those larval cells and allowed the mites to feed on that developing pupae for 8 days. What ended up happening was when the mites were removed and we did qPCR on them, so we tested for which viruses were in them and the pupa that they fed upon, essentially that orange mite became a blue mite. It acquired the genetically modified virus and was able to transmit the virus to another pupal host. So we're actually showing that mites can be dirty hypodermic needles inside of pupal cells. This has been long assumed, but never actually shown. We also showed that mites actively move from one adult bee to another to feed, and they actually do it at really high frequencies. And when mites return to a bee that's already been fed upon, or two mites go to that bee, they can acquire a virus as well. Now, we're bringing this all up because we really want beekeepers to understand that as mites are increasing in numbers inside of your colonies and they're actively feeding on both brood and adult bees, there's consequences to that continued vectoring and viral circulation within a honeybee colony. Now, viruses are going to be transmitted in a honeybee colony not just from adult bees cannibalizing pupae or from mites feeding, but also from bees engaging in trophylaxis with other adult bees. So if you're not familiar, trophylaxis is the oral exchange of food between two individuals, and honeybees engage in this behavior all the time. They do it for both communication. They have this thing called the social gut. They essentially need to know how much protein is circulating in the honeybee colony. So a lot of the trophylaxis are these really short events just for communication. But older forager bees also need food donations of amalgamated protein, and the nurse bees provide this to them. So sometimes bees will engage in trophylaxis and the event will take a while. One bee will open up its mandibles and another bee will stick out its tongue. The bee that's opening up its mandibles is the donor bee. The bee that's sticking out its tongue is the recipient bee, and it's essentially getting protein from the younger bee. Now, this event is an opportunity for oral transmission of a virus. And we have already known that viruses can be transmitted orally in a honeybee colony. And during my thesis, I wanted to take this a step further and ask just a really important question, which is, can trophylaxis act as a bridge maintaining deformed wing virus in the host population? And can mites acquire that virus by biting bees which never came in contact with the original infectious agent? And we have this schematic right here. So over here in A, we got some mites and got the genetically modified virus inside of them. And we had them feed on adult bees, which is depicted right here with the letter B. And these were in special cages that I designed that had 2 compartments and a screen in the center. And the screen allowed the bees to engage in trophylaxis with each other. We removed these mites, so there was no risk of having the mites in the experimental arena. And we provided recipient bees over here in C. And they co-housed with these BBs, that's a weird way to say it, through the screen for 11 days. And they're engaging in trophylaxis, they're sharing food. Well, we removed these bees over here, left the donors, and we added new mites, and these mites are naive. So we tested them ahead of time. They had no virus, no genetically modified virus in them. We had them feed on these adult bees that were recipients. And lo and behold, a low number of these mites acquired the virus and were able to transmit it to pupae. And so we really have, in the lab, what we're showing is essentially, if you think of a whole brood cycle with Varroa, on a larva emerging, feeding on a bee, those bees trophylaxising in a honeybee colony, another mite coming on and feeding on that bee, and then going back into a brood cell, the virus can actually start with one mite and one pupae, go through a series of bees circulating in your honeybee colony, and end up with a very different mite with a very different pupae later on. And so trophylaxis can act as a bridge maintaining a deformed wing virus in your honeybee colony. Now, I really want to point how scary this is for honeybees because you're likely not giving your bees enough credit for what they're dealing with. Deformed wing virus is different than most infectious diseases on Earth. So most infectious diseases are either vector-borne or transmitted host to host, but they're not both. And so what do I mean by this? Well, we're all familiar with the plague. It's caused by a pathogen called Yersinia pestis. And in Yersinia pestis, that causative agent for the plague, it can be transmitted by the biting of fleas or lice coming off of zoonotic coming off of rats and biting people. So if I get bit by a flea that has plague in it, essentially, I get bubonic plague. It's vectored. Now, if I cough that pathogen into any other human here, I successfully transmitted the pathogen, Yersinia pestis, host to host, a horizontal route without a vector. And you would get pneumonic plague. And we've most likely all heard these terms before, bubonic and pneumonic plague. They describe the transmission route at which someone acquires that pathogen. Now, most infectious diseases on Earth aren't like this, not at all. So let's take something like malaria, which I actually modeled a lot of my honeybee research off from. Malaria is transmitted by the biting of an infectious mosquito. That's it. If I get bit by an infectious mosquito, I could potentially acquire malaria. But as hard as I try, I can't cough malaria into anyone. It's just not a viable transmission route for that pathogen. On the other hand, if I showed up to this meeting in real person, I had tuberculosis, I could totally share that host to host, person to person, and you all could acquire tuberculosis. But it doesn't matter how many mosquitoes I put in the room with us, none of them could bite us and then acquire tuberculosis. It's not a transmission route, it's not vector borne. There's actually an extremely few number of infectious diseases on Earth that are both vector borne and communicably transmitted host to host. Yersinia pestis is one. It caused 3 pandemics in human history, suppressing up to 50% of the human population in Europe in the Middle Ages. Deformed wing virus is another one, and some of the other viruses in honeybee colonies, but not all viruses in honeybee colonies. And then a few other emerging pathogens here on Earth. One of them is called Tembusu virus, West Nile. Salmon isovirus is another one. Japanese encephalitis virus is another. And then I don't know why, but my brain is actually skipping on the other one right now. But the scary thing about these pathogens is they went from relative obscurity in the 1950s, just like deformed wing virus, and they're now expanding globally across the Earth. They all suppress the host on the individual level, so they can kill the host, or they can suppress the host on the population level, meaning if it circulates enough within your host population, you can have a complete crash. And these pathogens, like deformed wing virus, actually thrive in unnaturally dense settings. And I want you to think about what your honeybee colony is like, which is it's an unnaturally dense environment. We normally don't have 10 beehives right side by side, not of Apis mellifera in North America. They naturally spread themselves out, as Tom Seeley has shown. And it's a successful strategy for mitigating pathogen transmission from colony to colony. Well, think of what the plague did to humans in medieval London. It's unnaturally dense. It just wiped people out. Like, you were far better off being either a broke farmer in the countryside or rich and wealthy and isolated in the countryside. Just like an isolated beehive is far better off. Same thing with salmon isovirus. It can cause 100% losses in these unnaturally dense farmed factory cages of salmon. Same thing with Tembusu virus with ducks in Southeast Asia. It's responsible for the death of hundreds of millions of ducks. So when I say we're not giving our bees enough credit for what they're dealing with, they're literally dealing with a plague of honeybees, and we don't necessarily acknowledge how difficult it is for a honeybee colony to survive if it's dealing with essentially plague transmission dynamics. And so at the end of this, I say, is there anything good a mite's doing inside of your honeybee colony? And on a short time frame, the answer is no. They shorten the lives of your bees when they feed on them during development, and they cause deformity. They shorten the lives of adult bees. My research, which was just recently published, clearly shows this. They directly vector viruses. And not only that, once they vector viruses, that adult bee could be a super spreader transmitting from host to host. They also reduce the colony's ability to successfully forage or rear brood well. 1 second, I need to cough. And so there's nothing great that mites are doing on your worker bees. And as scary as everything I just shared is about mites, most of this is not happening in your honeybee colonies during the early part of the season. So essentially now in the spring, through early summer when you have your main flow. And that's because for the last 30 years, we got an extremely important piece of information about Varroa destructor completely wrong. And I'm going to give you a spoiler alert on the next slide. So for the last 30 years, we've told everyone that mites prefer nurse bees. This is something that you've probably heard in your beginning beekeeper classes, and it's what all research has told us. My research overturns about 30 years of honeybee Varroa research because we found out this is absolutely not true. Mites prefer and aggregate on adult drones at extremely high levels. And in fact, they only shift onto worker bees when drones seasonally disappear from your honeybee colony. So the rest of this talk now is going to talk about the consequences of this new finding and the consequences of what happens in the middle of the summer when your drones disappear and might start to feed on your worker bees, which all of the transmission dynamics I just shared with you for the last 10 minutes then kick into place. So historically, why have we thought mites prefer nurse bees? Well, some of our earlier research on Varroa were simply descriptive studies. And in the early 80s out of South America, there were some descriptive studies of Varroa, and they described them on nurse bees or worker bees. And some of our earliest work in the laboratory did host preferences of Varroa, but these experiments, all of them here, were all done in the laboratory except for the last one. And every single one of them, including the last one, completely excluded drones. They looked at mite preference on newly emerged workers, nurse bees, or foragers. So they were looking at that one cohort, the female cohort, and ignored the drone cohort. And so all of our research has been on worker bees, or largely on worker bees. And as a result, a lot of our beekeeping management has focused on worker bees instead of mites on drones. And so my work quickly asked, is this correct? And so what I ended up doing was I set up these really simple laboratory trials where I had paint-marked drones and paint-marked nurse bees. I put them into these modified petri dishes with a feeder, and I added 10 mites per cage. And I did 3 trials. The first 2 trials, when I compared young drones to nurse bees, mites overwhelmingly aggregated on the drones. I did a 3rd trial just testing drones and workers at the same ages, and I ended up getting no difference in the distribution of mites between workers and drones when I did that. I thought that was interesting, but what I wanted to really do is get directly out into a field trial. We do so much of our beekeeping work in the laboratory, and what's really important is seeing, are these behaviors of this really devastating parasite something that we can also see out in real colonies? So what I ended up doing was setting up a tedious but relatively simple experiment where I wanted to be able to test in real colonies whether mites aggregated or preferred adult drones over worker bees. So in short, what I did was I marked bees at emergence for 5 continuous days. And I used one unique color for each day. And when I do that, I now am able to identify bees and their age based upon the color that's painted right on the center of their thorax. So let's say this was Monday and it's day 1 of the experiment. If you emerged, whether you're a drone or a worker, you're going to be painted blue. If you emerge with no mite, I pick you up, I check you, then you just stay blue, and I'll have tons of blue drones and tons of blue workers. But if you emerged with a mite, I mark you blue, I know your age, but I'll give you a second color, in this case, red, and I'll put it on part of your body where I won't reuse that color and location with any other bee in the colony. And I'm writing this all down on a field sheet, on a notebook. I put all the bees back in the colony, and I go back inside. And when I started this, it was March and April 2020. So we just entered the pandemic and lockdown. So I essentially did this experiment and then went inside and watched Tiger Kings. You know, it was really tacky and not the best part of my life, but what are you going to do? No one could predict a pandemic. So on day 2, I would mark all the newly emerged bees yellow. I'm not showing that here just to kind of keep the schematic clean, but I would recheck all of Monday's blue bees. And if you didn't have a mite on you, you just stayed blue. I put you back in the colony. But if you gained a mite, I would remark you another color, write it in the field notebook, and not use that color for any other colony. And so I did this for 9 consecutive days. I marked for 5 days, and I rechecked and marked adult bees for the additional 4 days. I used a factorial design so I could remove redundant use of colors and reduce identification errors. And what I was able to do was answer a simple question, which is, do mites prefer drones or worker bees? I will just give us some background information right now. So 9 days was pretty specific for a couple reasons. One, it's hard to get more than 9 days of good weather in a row in Maryland. So stopping at 9 days was really important. We didn't want to have any missed days because of thunderstorms or excessive heat or rain. 9 days also covers most of the nurse bee stage, so we did that. The second thing is bees get really stingy as they get older. So we're losing a lot of our samples either by drones everting in our hands, which is annoying, or from nurse bees stinging us as they get older. So stopping at day 9 was just practical. And the bees really start to drift as they get older. So we're finding our samples in a bunch of different colonies. And we just decided for the sake of this study and to do a proof of concept, we're going to stop at day 9. This is what my colony looks like over on the right. It looks like a Christmas tree. You know, we have paint marked drones and paint marked workers, and we have non-paint marked bees. And I can go into my field notebook and tell you this drone right here with the green thorax and the blue shoulder with the green butt, exactly what its parasite history is. We did this rain or shine. I would absolutely not miss a day during the trial. So this is someone that helped me. This is Abu. He helped me in the second year of this work. And there was a rainy day and I was like, not thundering, we're still going in. And we just set up a pop-up tent and have some coffee and do the trial. So the next slide is the data from, sorry, I'm going to go back one. It stalled and then went too fast on me. So this is the data from 2 years of trials. There's 10 trials on 5 colonies. So we did 2 trials per colony. Over on the left, these are my early season trials. Over on the right, these are my later season trials. Early season trials, drones are available. So this really represents end of May, June, July. Over here, my late season trials, this really represents July and August. We have a very predictable severe dearth here in Maryland, and during our severe dearth, colonies will either slow down or completely cease drone production. So how to read this? Over here on the left, my x-axis is the age of the individual bee that I'm sampling, so it goes 1 through 9. My y-axis is the number of mites I observed on that individual bee during the inspection. There's 2 colors here. Girls are orange, boys are green. And so age 1, that essentially means the bee emerged with a mite on it. Whereas age 2 onward, they most likely gained a mite. A mite moved onto a bee that previously did not have a mite. As I said before, mites are really mobile, moving around inside of your honeybee colony. And so we can pick out a pretty clear pattern here, which is our younger bees are the bees that are most likely to have a mite on them. So this whole row is zeros. These are bees without mites at all. And up here, this is where we are heavily infested. Our heaviest infestations are on days 2 and then 3 after emergence. And drones are harboring most of the mites in these trials. In fact, it's actually a very small number of drones are responsible for harboring a majority of the parasites in these studies. Now, approximately 30 days later from every trial, of these early season trials, we redid the trial as a later season trial when drones seasonally disappeared. And now what's happened is the mites have shifted from drones onto workers simply because young drones are no longer available in the colony. We would still read this as the age of the bee on the x-axis, and on this vertical axis, the number of mites on each individual bee. Bees are emerging with mites, but the majority of the mites are actually moving on to bees that are 2 days old or 3 days old. And the commonality between this left over here and this right graph over here is the mites seem to love 2-day-old bees. 2 and 3-day-old bees, they move on to them at really high numbers. This right here is just a bar graph, which is the same thing as this graph over here, except what I'm doing is I'm showing you into bar graph form so you can get an idea of how many more mites are actually feeding on your drones, your adult drones, versus your workers early in the season. So they're harboring the most of those mites. Now, during my trial, I get some cool data, and I'm going to show you that next. And that is, I have some bees that go through the whole 9 days and we never see a mite on them. So those bees are never parasitized, or we'd call them a non-parasitized bee. We also have some bees that emerged with a mite, so we know that they were fed upon during larval development. We can kind of think of those as this green bee right here. And then we also have some bees that weren't fed upon during development, but acquired a mite at some point during their life. And these would be called parasitized during adulthood. And there's very few bees that were actually parasitized during both, but we can ignore them right now because they're such a small number. And so if I had a mite-free colony, this is what my colony would look like. And this is what every honeybee colony, Apis mellifera colony, looked like before Varroa destructor successfully jumped from Apis cerana onto the European honeybee. It was a bunch of non-parasitized bees trophylaxising with, or engaging in trophylaxis with other non-parasitized bees, taking care of the queen, rearing brood. All non-parasitized, really low chance of transmitting any viruses, which really weren't represented well in our honeybee colonies prior to the advent of Varroa destructor. But the moment Varroa jumps onto our honeybee colonies, this is what our colonies start to look like. They're no longer non-parasitized bees. There's going to be a mixture of bees parasitized during development and adulthood. Now, this is experimental colony number 5 that the early season trial, which is right here, was done on July 5th. So this is a pretty robust colony. And the relative parasite burden, the other way to say that, the alcohol wash, was 5.1%. So that is really high, right? We're already above economic threshold. And what I mean by relative parasite burden is when I do an alcohol wash, I get the number of mites over the number of bees in that sample. That's all an alcohol wash means. So if I get 5 mites and 100 bees, my relative parasite burden is 5 mites were on 100 bees. That's really different than absolute parasite burden. So my absolute parasite burden is something different. And that is, of these 100 bees, how many of them were impacted by a mite during the study? And that's what you're seeing right here. So at the end of my 9 days, I can count up how many of my worker bees at any one point during the trial had a mite on it and was being fed upon. And it turns out it was over 14% of my worker bees were bit during the trial. Now, this is actually extremely low compared to the number of drones which were parasitized during the trial. So 65% of the drones were bit during this trial. So there were a lot of drones that were studied, and 2/3 of them were parasitized during the study. Whereas a minority of your worker bees, when drones are present in the colony, actually get bit. So over here, a majority of these bees, of these worker bees, are still not parasitized when drones are present. Now, I said earlier, we redid every single colony with a second trial during the dearth when drones seasonally disappear. So 31 days later, I did that same experiment on this colony again, but this time drones are no longer really present in the colony. They're still there. There's a lot of older drones on the outside frames. There's a lot of older drones still going out on mating flights, and there's still some emerging drones, just not nearly as many as there were before. Again, when we get that dearth, it's pretty severe. So now my relative parasite burden, or my alcohol wash, jumps up to 14.6%, which is what is typical in the Northeast and Mid-Atlantic. We really see these jumps in August in our alcohol washes. And a lot of people have said these are unexplainable. And we've come up with a lot of different hypotheses of why would the alcohol wash suddenly spike that much. But my effective parasite burden, remember, that's how many bees at one during the trial were actually burdened by a mite, not just how many happened to have a mite on them when I sampled. Very different things. Actually jumps up to almost 60%. So within 31 days, I go from this, which is a minority of my worker bees are parasitized, this, where a majority of my bees are parasitized in 9 days. So right now, if I'm a non-parasitized bee, one of these brown bees, I'm surrounded by parasitized bees. It's almost impossible for me to engage in colony-related tasks without having to interact with a nestmate who's going to be parasitized by Varroa, which means my chance of acquiring virus skyrockets. And I really want to point out 60% of these worker bees were bit in 9 days. So it's a really short period of time that these mobile mites in your colony, once drones disappear, can just devastate your worker bee population. And this is the most nefarious thing that I think Varroa are doing to our honeybee colonies and to us as beekeepers, which is this colony didn't look that bad. This is what 60% bit looks like, okay? It's really not that bad, unless if I did an extremely close inspection here. It's got a beard on the entrance on the bottom board. If I open it up, it's covered in bees. The brood really doesn't look that bad at first glance. You can see some spotting where they're uncapping and removing things, but there's no real pathology that I'm nervous about. And the honey supers are still full. They're still bringing in pollen. They're still bringing in nectar. This colony, at first glance, is really telling me I'm okay. And this is the most nefarious thing to me, because this picture right here is actually this picture. It's 60% parasitized by Varroa. I just don't see the collapse of this colony for weeks later. But it's essentially already walking dead, because once Varroa bite those bees, you can't unbite the bee. I can't get unbit by a zombie. Those bees can't get unbit by a mite. So going forward, even if I was to treat on the last day of this trial, 60% of my bees have already been parasitized. This is what another colony looked like in the trial. And again, this is just the brood nest here. Honey supers have been removed. And ostensibly, this looks like a semi-decent colony that you would probably just feed and treat to try to get it through the winter. Some people in August would definitely try to make some nukes out of this and rear them up. And our research is saying that there's a huge risk to trying to do this with your colonies, because you can't see which bees have been parasitized. They don't glow in the dark like my schematic did. Now, conceptually, my colony looks like this early in the season, where there's a lot of mites on the drones, both in the brood on emerging drones and on adult drones early in the season. And there's relatively few mites on the massive number of worker bees early in the season. And what's happening here, our drones are providing a cushion to the effects of parasite burden on your honeybee colony. Drones don't engage in brood rearing behavior. They don't take care of the queen. When they engage in trophylaxis with another bee, they only receive food. They don't donate. They are extremely poor candidates to transmit viruses within your honeybee colony. And mites are feeding on them right now, spring, all the way through your main flow. They're providing a cushion to the effects of parasite burden. But the moment drones seasonally disappear, it's like Hoover Dam is busted open, and the mites shift onto your worker bee population. Now think of it this way. Your worker bees, once they're parasitized, they still rear brood, they still take care of the queen, and they still engage in trophylaxis with the rest of your nestmates. The cushion is completely gone, and your colony starts to bear the impacts of Varroa destructor. Now, we wanted to see if these trends were observable when we picked colonies at random and when we selected bees at random in colonies across the season. So I used to work for Michael Palmer up at French Hill Apiaries, and Troy Hall in Vermont, New Hampshire is a close friend. And both of them allowed our group to go up there and sample their colonies for a season. And what we ended up doing was we picked 48 colonies in 8 apiaries at random. And we wanted to see a couple things. One was, is this trend that we just showed in experiments in Maryland observable in a different location and at random in random colonies? That was really important to us. The second was, we wanted to see if there was some sort of predictive value with sampling drones early in the season. And so what we ended up doing was we came up with a new sampling technique, and we paired it with an alcohol wash. And I'm going to walk you through that method and that data right now. So what we ended up doing was we chose a colony, and we did an alcohol wash on it right from the center of the brood nest. And then what we ended up doing was we hand picked up 100 worker bees and inspected them for mites. And then we hand picked up approximately 40 drones and inspected them for mites. So we essentially did an alcohol wash, and then we kind of did an alcohol wash by hand. And this is the data that we got from this. Okay, so one sec, I gotta go back. Okay, so this is the hand alcohol wash, hand picking up drones and workers. So my x-axis, instead of being days this time, is months. So May, June, July, and August. My y-axis is the number of mites that were on an individual bee. So these are all individual bees, every single dot here. And if they're down here in these big blocks, they were an individual bee that had no mite observed on it, which most of the bees that when we hand pick them up shouldn't have a mite on them. And if you are a male, you're green, same color. And if you're a female, you're yellow, you're orange. So in May, we had very few detections when we're hand picking up bees. And most of those detections were on drones. All of our multiple detections were on drones. Come June, we have an increase in the number of individual bees with a mite on them, but it's still really low. We get into July. Now this is up in Vermont. Our main flow, basswood, sweet clover is hitting. We just finished up with staghorn sumac. The flows are really heavy. Colonies are in peak production for drones, and we see a huge spike on the number of detections. And again, these detections are predominantly on drones. We get our first multiple detections where there's more than one mite on worker bees for the first time. And then we get into August, and all of a sudden, mites don't prefer drones anymore. Well, we know that's not true. What happened in August? Well, even though we have a pretty good fall flow, drone production plummets in August. We're entering a dearth up there. We still have drones, just not nearly as many as we had July. And we're already seeing that shift of Varroa destructor from drones onto worker bees when we do this hand sampling. And so that's what we're seeing here in this pattern in August. So we have almost as many detections as we did in July, but those detections are predominantly showing up in worker bees now, not because mites no longer prefer drones, but simply because there's not that many drones in the colonies anymore. Now, I said we paired this work with an alcohol wash. So this is our alcohol wash data, and the pattern follows the same as our hand wash data. We have very few detections early in the season, and all of those detections are low. We have more detections come June and July. And all of a sudden, when drones seasonally disappear from our colonies, we get a spike in the number of detections and the level of which detections are occurring in alcohol washes. And our argument here is this is when alcohol washes are finally doing their job. They're actually sampling for what they're supposed to be sampling for. And that's what I'm going to talk about next. Are alcohol washes even sampling for what we're asking them to sample for? Now, remember, early in the season, I said conceptually, our colony looks like this over on the left. And late in the season, our colony conceptually looks like this over on the right. So over on the left, few mites, predominantly on drones. Workers are pretty much mite-free. That's a generalization. Every colony is going to be different depending on its load and distribution. Late in the season, we have very few drones. Lots of mites now on a smaller worker bee population. Again, a generalization, but work with me here. And so what we're asking for is, can an alcohol wash, which was designed to collect nurse bees, actually account for a shift in the distribution of Varroa over time? And this is what I mean by the shifted distribution, where we go from distributed on drones to distributed on worker bees. And the easiest way for me to explain this is by using M&Ms. So when I first proposed this to my committee, it was during the initial lockdown of the pandemic. And what I ended up doing was going to Costco and buying some M&Ms so I could make a visual display to explain to my committee. And I actually ate 80% of this container in like half an hour while watching Tiger Kings. It was really pathetic. So I went back out to Costco, I bought another container, and I laid out all the orange M&Ms because for some reason I did not get a container that had a lot of yellows. And these are my bees, okay? I guess you can ignore the jokes. These are my bees, the orange M&Ms. And my blue M&Ms are my mites. If we assume that mites were uniformly or maybe randomly distributed across our worker bee population, then the picture on the left would be how mites should be distributed in our honeybee colony. And that's not the case, because we know from my work that mites are actually aggregating on a small cohort of male bees, or even aggregated within that cohort by clumping onto certain individual drones. So their distribution actually looks more like this over on the right. So what's the consequence for an alcohol wash? Well, this is what I showed my committee. If I scoop up 300 bees and I miss my clumps, then I get a 0 for an alcohol wash early in the season. And we definitely know they're not zeros, they're no detections. You simply didn't get a detection. But if I get a clump, I might get an artificially high number. And this is the problem where we try to use a sampling methodology that was designed for random distribution on a parasite that actually distributes in an aggregated distribution. So we brought this up as an issue for the alcohol wash, but now we're trying to provide a solution for beekeepers, especially hobbyist beekeepers. So what I proposed was that we could use targeted sampling to identify colonies that are going to have mites early in the season, earlier than we normally could. Now, targeted sampling is a sampling methodology that's used when you have a disease that expresses itself at really low frequencies in your population. It means you can't just randomly survey people to find out who has the disease. Or it's used for diseases or behaviors that have a taboo, right? So think of like in the 80s, if you had HIV, it was super taboo. You couldn't just randomly call up 30,000 people and ask them if they had HIV. You would get lies all the time. So what they did was they would find groups, a few people that had either that behavior or that disease, and then ask them to self-refer, to refer someone within their own network, and that's called targeted sampling. And so we looked at that and we said, hey, we can modify this biased sampling technique and use it for Varroa and possibly detect mites earlier than we normally could. So we're going to modify this thing that's used for doing disease surveys and use it for our own parasite and honeybee colonies. And instead of trying to create a sampling methodology that's unbiased, we're actually going to make a really biased sampling methodology. And that essentially means that we are going to go ahead and ignore trying to collect lots of worker bees and just go right for the drones. And so this is probably like just a really good picture of what we mean, which is instead of trying to sample 300 worker bees and killing them to find a mite or two, and then really not knowing what to do with that data early in the season, we're going to sample just a handful of drones and see if we can get better quality information from those drones while creating a sampling methodology that's quicker and sacrifices fewer bees. We're in the process of doing this. And so essentially, I want to know, can sampling here over on the left early in the season predict this over on the right later in the season? And the thing I really want to point out to everyone is the colonies that become heavily infested will really surprise you. So we're going into colonies at random, and we're actually excluding colonies, though, that already have an underlying disease. So if you have chalk brood or EFB, we want to exclude you. You know, we don't want to try to do try to prove a sampling technique with confounders right now. But some of our healthiest looking colonies were actually the ones that had really high mite levels at the end of the season. And arguably, it's because these are the best colonies that are breeding reservoirs for Varroa. They produce massive amounts of drones, and those massive amounts of drones provide a cushion for the effects of parasitism. So mites can reproduce at really high levels and not hurt the colony early in the season. We're still doing the data on this, but I'll share a glimpse of it right now, which is this bar graph up here. It's just descriptive stats. There's no analysis really on it. My blue bars are the percentage of drones that had a detection in May, whereas my orange bars are the number of mites, or the percentage of mites in an alcohol wash come July. So blue is early in the season on drones, orange is later in the season alcohol wash. And we see this pretty strong pattern, which is colonies that have high levels of detections early in the season on drones produce colonies, or have high detections in the alcohol wash later in the season. And here's what we really want to bring up about the alcohol wash, which is we don't really care in the fall if you are telling me with an alcohol wash that my house is already on fire, right? So if I get a really high number in my alcohol wash late in the season, I can't do that much with that information, right? You're essentially telling me I need to immediately treat. And my research is already saying if you have a high alcohol wash count, then a lot of your worker bees are parasitized, right? And I can't undo those mites even if I immediately treat. So what's much more useful to me is having some sort of way to predict which colonies are going to become heavily infested in the fall. And so we're trying to come up with a method right now where you simply hand pick up the drones. This picture is on here, not just because it's a good picture, but for a really important reason, because this colony was essentially a textbook example of the issues that we had here. This colony looked gorgeous. Everyone liked to take pictures with it in the season, but it was essentially a brood factory for Varroa. And all of our detections early in the season were on the drones. But come August, when the colony is naturally getting a little weaker, and the drones aren't being produced, the brood was just pocked with Varroa. And we were just constantly picking up worker bees with mites on them. So this colony produced massive amounts of Varroa when it was super healthy by having a huge, robust drone population. And those drones provided a cushion, kind of not allowing the mite parasitism to hurt the colony. But when that main flow ended and the drone production ceased, man, that Varroa just shifted onto the worker bees. We saw a huge spike in the alcohol wash, and the worker bees are just getting bit like you wouldn't believe. But that alcohol wash in August isn't that helpful for Michael Palmer, right? We're essentially telling him, hey, by the way, this colony is now heavily infested. And again, my research says it's not just that you're going to get a high alcohol wash, but 4 to 6 times as many bees are going to be bit by Varroa. And so this is where we're really trying to encourage some earlier detections and for people to take action earlier on. This is the end of my talk, and I hope I left enough time for questions for a Q&A. I first have to give out some thank yous. During the second year of this work, I had some amazing assistance from a couple interns, undergraduate helpers, and it was super appreciated. While I was doing this work in Vermont, they were holding down the fort for me in Maryland. Jay Evans is now in his lab at the USDA, but he has been unbelievably helpful and supportive of all of my work. And this is going to be the dorkiest thing that I share next, which is I didn't have funding for the Vermont work. And so I called my parents, and they got up once a month at 3 a.m. and drove from Eastern Connecticut to Vermont, sampled bees with me for 3 days, and then went right back to work on Monday. And the project wouldn't have been possible without them. And they did this for 2 years. So we just finished our second year of work this summer, slowly processing the data, and we're really hoping to put out some good information for everyone. In this picture, my girlfriend at the time was super supportive and awesome, joining on these adventures, and a beekeeper friend in Massachusetts helped out for 1 month, which was really great. This is my actual email address, so you can write it down. And oh, it's funny, my mom just texted me. If you want to get in contact with me. And then for what's next with this work, I'm doing a citizen science project with members in Virginia with James Wilson, who's an extension specialist out of Vermont Tech. And so with approximately 60 people and a couple hundred beehives now in a different region, we're going to see if this predictive drone sampling can work, and if we can start to change the paradigm of how we approach Varroa and manage Varroa. But at this point, I will stop my screen share, and opening this up for Q&A.
Thanks, Zach. Really excellent presentation. I'm very grateful for the data that you've shared with us. A lot of questions popped up in the Q&A. I am submitting my resignation for beekeeping here at this moment. I quit. All hope is lost. These bees are just under a total onslaught.
Well, let me respond to that right away, because we've had a paradigm for decades, which has kind of been a bifurcated paradigm. We have one group that essentially wants to be treatment-free and have the problem solve itself, which it's a really complex problem, and on the human lifespan, it actually might not resolve itself. And what I mean by that is we have to give credit for how complex this parasite is. Not only, I mean, it's an evolutionary host jump for one. So for millennia, honeybees have existed, and now within 50 years, we have a new parasite on them. So that's very different, right? Not only that, but we've had these honeybee viruses that have been in our honeybee populations at really low levels. And now suddenly we have a parasite that can vector them, and we saw this massive amplification of those viruses. Varroa could have jumped onto the honeybee population and never vectored a virus. That's totally possible, but it turns out it's a competent vector for a lot of honeybee viruses. So now your honeybees, which have co-evolved with these viruses to deal with oral transmission, contact, vertical transmission, all of a sudden they have to deal with vectoring of those viruses, and they're not evolved to deal with that. So when we talk about mite resistance, it's relatively vague because there's a lot of nuance in that conversation, and it's hard to have it when we try to define something as a single term. So I'm not dissing treatment-free beekeeping. I actually think it's part of the solution. Don't get me wrong. I actually think it's part of the solution, but it's a much more nuanced conversation than we're often allowing ourselves to have. The other part of that paradigm has been to treat. And treating has followed the paradigm most often of super my honeybee colonies, get my production out of those colonies, remove the honey supers, and try to treat before my colony dies. So that's the paradigm. In both cases, I have, whether I'm treatment-free or I'm following a treatment paradigm where I super, get production, and then try to treat, I have my honeybees living with a high number of mites and what can be a large circulation of honeybee viruses. There's a different paradigm we can engage in, and what I'm really proposing is predictive sampling and removal. So let's think of it this way: there's 80 people, so nice round number, in this participating in this right now. Let's say we were all in this room together, right? And let's say I had tuberculosis. Okay, I'll be the index case. So a really successful disease strategy isn't to give me antibiotic and let me continue circulating with all of you. Some of you are going to get sick. Okay, there's no way around it. A better disease management strategy is simply to remove me from the population. Okay, so you can do that by having me isolate at home, or you can have it in a different way where you test and simply don't allow me in. Well, we can do that with our honeybee colonies. Let's say early in the spring, so it's March here, we can get into our colonies here in Maryland. I can go in and do my drone sampling on a bunch of honeybee colonies. Most of them are going to have low or no mites detected on the drones right now. That's great. But I'm going to get a handful of them, of the 60 or so colonies I have, they're going to have multiple detections on the drones. Now, my current paradigm is to super them and get production out of them. Well, that means I have infested colonies in the same community as my non-infested colonies. That's the equivalent of having me show up at tuberculosis and hang out with y'all. What if I just removed those colonies? And what I propose is take those colonies with the drone detections and break them down into either nukes or split them, treat them, and then requeen them. Now I have more colonies going through the season that are mite-free, and they're not co-housing essentially in the same apiary with my low infested or mite-free colonies. What I'm probably doing is conferring protection to all of my non-infested colonies simply by doing removal. And I actually think this can work with treatment-free beekeeping. And the reason being is I actually want to find queens that are naturally good at suppressing mite reproduction through whatever mechanism that is, whether it's uncapping or just making really weak pupa that die as soon as mites feed on them, whatever it is, I don't care. But if mites are reproducing at high levels on your drone brood, you, as a queen, you've essentially already told me I'm not capable of suppressing mite reproduction. So if I am interested in finding successful queens, I want to remove that queen from my community, but I want to give the new queen that I want to test out a clean slate. You know, we essentially wouldn't want to throw her into a nuclear disaster. So instead of throwing my hands up saying, I want to quit beekeeping or this is a huge struggle, I actually think the predominant thing we need to do is switch the paradigm. And we haven't given ourselves an opportunity to necessarily do that yet. And I say that unfortunately because we have really good beekeepers now. Whether you're treatment-free or treating, whether you're commercial or a hobbyist, you can't be a bad beekeeper and successfully keep bees year to year anymore. It's just gotten too difficult. There are too many multiple stressors on honeybee colonies, and mites are actually seemingly becoming more virulent. So you can't be a bad beekeeper and keep bees anymore. You know, if you do, you're on the hamster wheel of buying packages every year. But I just think we haven't tried the new paradigm yet. So I'll halt, so I guess I can read some of these questions now, James, huh?
Yeah, so selection works against the beekeeper too, huh?
Mm-hmm. Yeah, so hey, this first question by Anthony is a really good question. The question is, obviously a bee with deformed wing virus will never become a forager, but how long can they be beneficial to a hive in performing general hive tasks as they age? Or are they not that functional as bee? So let's just kind of stop there because we get into multiple things. So bees with deformed wings are extremely short-lived and they won't engage in many or any hive-related tasks. They'll definitely beg for food. And bees will actually lick them at really high rates, which is weird. They're a really poor candidate to be a maintenance host for deformed wing virus. And let me use this as an example. So let's say we go back in medieval times and I get that plague in me, you know, a flea bites on me and bites me, right? And I'm walking towards our castle. If I die within a day of being bit and die like out in a field, you're all protected, you're all safe, right? Because I died so quickly, I had very little contact with the rest of my village. So you're all protected. Same thing in a honeybee colony. Those bees are actually pretty poor candidates to be maintenance hosts. In our experiments, some bees could be bit and live for 15 days and develop high levels of virus. Those are really good candidates to transmit a lot of virus in the honeybee colony because they're still engaging in hive tasks. They're still communicating with all the other bees and interacting with them. And they have 15 days of contact with those nest mates. So they can cause a potential super spreader event. So there's this disconnect we have where the symptomatic bee is extremely visible to the beekeeper. The asymptomatic bee is not visible, but can actually be highly infectious. So we kind of have to uncouple what we think about which bee is actually deadly in a honeybee colony, because you can't see the ones that are actually causing a lot of transmission. Do you want to pick out questions or do you want me just to go through?
Yeah, I can. I have a couple that I had picked out.
Go right ahead.
And I'm riding the roller coaster here. So I was going to tell you that, you know, I'm a licensed clinician and I can help you with your Tiger King trauma, but I might have to do some self-talking here because I'm riding the roller coaster of I quit. Okay, I'll beekeep. Okay, I quit. You know, lots of good information though, and it just really helps paint a clearer picture of the complexity that we're facing when it comes to deformed wing virus or viruses in general and the way that they're vectored and spread, and that it's just not so clear cut when it comes to either treating or not treating. And we really, like you said, have to be better beekeepers. If you want to breed better bees, regardless of your treatment paradigms, you've really got to be focusing on a lot of these nuances. It just so happens that beekeeping is so addictive. I think a lot of people are committed to kind of working their way through that. And like I mentioned earlier, you know, you kind of get selected out of the process because you can't engage with beekeeping anymore and be successful haphazardly. Like, you really got to be focused at it. So one of the questions that I wanted to bring up, this is one of my questions. I don't know if you're familiar with any of his work. Dr. Ralph Buchler from Germany. He's done a lot of work on sustainability and beekeeping. I think he's worked out of Kirchain for a while, but he had proposed just recently before his retirement a method regarding cohabitation with Varroa, and that he would suggest that you do some, a brood break and some drone culling throughout the course of the season, or engage in a queen caging to reduce their reproduction. But at that point, if the levels were too high or reaching a specific threshold that they had set in their study, they would do a summer treatment. But at that point, after the summer treatment, there was no further treatment in the winter, and they would allow the colony to winter with the Varroa load that they had. And they come into the spring, and then that way the drone, and this is the theory, is that, and I think they had practiced this and showed some efficacy, the theory is that the drones that are parasitized aren't able to go on and reproduce, and therefore are kind of selected out of the process, allowing the drones that are able to endure these infestations, or the virility of the mites, are able to go on and reproduce with queens, and maybe that's passing on some resistance through the heredity. Does that make sense? The question makes sense. Do you see that as possible?
I think there's a lot there to uncouple, and I like to kind of keep the question super simple, straightforward, instead of opening up something that becomes complex with a lot of confounders. So the last thing that you're mentioning there is essentially transgenerational immunity.
Right.
If I have a low level of a pathogen in one generation, does it confer some immunity across generations after reproduction? And so there needs to be continued research in that. That's something our lab's interested in, but not necessarily anything I've worked on right now. Simply kind of, I think that's probably the best way to answer that. And I'm not super familiar with that person's research, so I'm not going to spend too much time answering questions on that.
So let's see. Are any of the viruses that infect and weaken bees a threat to the small hive beetles? It seems that they just are impervious to almost everything, and they're just obnoxiously.
Yeah, I don't know. I don't study how viruses possibly transmit or don't transmit to small hive beetles, and I think that's just a different management talk. Yeah.
Do we need to encourage drone production throughout the season through foundationless combs? This particular person does top bar hives, but would drone production become more of an attractant to the mites? And then if you were,
yeah, I get this question a lot. And so let's think about like what's going on in the honeybee colony, which is your colonies. A healthy colony is naturally going to want to produce drones. And why? Because if your colony doesn't successfully swarm, an evolutionary success story for your colony is a drone mates with a virgin queen not within your colony. So, whether you like to or not, your colonies want to produce drones. So the question now is, do we want to encourage even more drone production? Well, if you encourage more drone production and it already has a low level of mites early in the season, you're allowing those mites to reproduce at really high rates during the season, during your spring through main flow. And that's because mites reproduce at really high averages on drones versus worker broods. So essentially 3 to 1 every time they go through a cycle. So you're not going to notice the effects of that because it's largely being cushioned. Those drones aren't rearing brood or engaging in much trophylaxis. They only receive food. So you're not really going to notice it. I would say a way to reframe it is if you're going to encourage drone reproduction, then maybe engage in drone comb removal as a mite management strategy. Calderon has already shown that the intense removal of drone frames from a honeybee colony can successfully keep mite levels below economic thresholds. And this is like in places like Cuba where they don't have access to miticides. This is how they successfully manage for mites. And it can be a really effective strategy. It's not a silver bullet, but it's a really effective strategy. So instead of saying maybe should I encourage it, I would say, well, if you do encourage it, go the one step further and every 3 weeks essentially just remove drone frames.
Don't forget about them either, or you'll have drones all over the yard.
Yeah. Well, if you forget about them, you'll essentially be like that last colony that I mentioned, which is your reproductive factory for Varroa at that point.
Yeah. Yeah. Now you mentioned that the mites were aggregating on the drones. Are they also feeding on the drones? Maybe, and this question suggests, do they have more fat body to be feeding on? But further that question along, like they're aggregating there, but are they also feeding on the drones?
Yeah, so they are feeding on the drones, and that was one thing that our research showed, that they're not going there just as a resting spot, but they're actually going there to feed on the drones. So the work that we're doing this season is partially asking why are they feeding on the drones over worker bees. And so the nutritional component is the most likely parsimonious answer to that, which is if I feed on a host that is the most nutritious, then it improves my fecundity. And so that's our assumption, that they're feeding on the drones so they can actually be better parents, raise more offspring, is essentially the short way to put it. And so we'll go ahead and test that. The problem for beekeeping management is it doesn't necessarily help your beekeeping management, right? Like if I turn around and say, yes, they're actually feeding on drones because the drones have more fat body and they're more nutritious and all these things, it doesn't change how you keep your bees. So it's a really interesting topic for us, and possibly for creating non-traditional miticides or mite control strategies through some sort of feeding through drones. But it doesn't necessarily change your mite management.
And the viruses are going to equally vector through the trophylaxis occurring with the drones and the workers. They're not really, are they creating a sink for the virus?
Well, we don't know that yet. So that was something I did study, but there's a problem, which is drones defecate a lot inside of cages. So we're trying to study trophylaxis with drones and workers, and to see if an infectious drone, even though it's only receiving food, can still, through that contact, transmit a virus to a worker, because that's important information. And unfortunately, we had this confounder, which is the drones defecated in the cages. And so our virus prep is showing up on workers, whether or not the worker became infectious, simply because there's infectious fecal material on those workers. And we think it's just an artifact of our laboratory arena. Drones can be really good about holding essentially their poop and then going outside. But in the laboratory, it's a completely different arena, and they'll just defecate. So we don't have the answer on that. It's an extremely good question, whoever asked it.
So this is a question that came up for me, being being a treatment reluctant beekeeper. With what you're proposing, what your study is kind of revealing, it would, like, even for me, would encourage a prophylactic treatment in the spring, because you almost you're kind of, it's a ticking time bomb, so to speak. But would you recommend, or strongly recommend, treating with miticides in spring? And what does this new information mean for queen mating season, mating with parasitized drones?
Yeah. So yeah, we also tried to look at that. What's his name out of Purdue, who showed that there's so many dud drones? We didn't show it, but this is one of the proposals we're putting forward, which is essentially twice as many drones as you think are actually getting parasitized by mites. Is that one reason we have so many essentially dud drones? And that's for someone else to do that work. We tried to do it. Our N wasn't big enough. We lost a lot of our drones in a field trial. I'm probably not going to repeat it, but it's a really good question. Should you do a spring treatment? If you're a commercial beekeeper, you're already doing spring treatments. You have to. End of story. If you're a hobbyist and you have less than 10 hives, to be honest, if you can't do some sort of detective sampling in the spring to find out which colonies are harboring high levels of mites, then you're probably not putting enough effort into your beekeeping. So I would say do that first, and then consider your treatments. You can then choose whether you want to spot treat, just treat those ones that are infested, or if you want to prophylactically treat. But it's definitely not a bad idea. I now, in my apiaries, what I do is I go through and do my drone sampling. I hand pick up about 40 drones from every colony. And the ones that have multiple detections, I don't make nukes out of them. So any of my customers that get nukes from me, they're never from a parent colony that had multiple detections on the drones. And why? My hypothesis is I'm essentially giving them what looks like a really nice nuke, but it's essentially seeded with enough Varroa by the fall, they've got a problem. I don't want to do that to my customers. And then the second thing I'm doing is I'm treating all of those colonies with multiple detections early on. And this is really good for me because I do honeybee research. My ultimate goal is to figure out how are pesticides affecting honeybee colonies. It's really hard to answer that question when there's so much Varroa, because the symptoms of mites and viruses can really look like pesticides at sometimes. And so we have to uncouple them so that we can really understand how pesticides are affecting honeybee colonies. But I would go right back to spring treatments are never a bad idea. I think if you have a few colonies, you can do one step better, which is instead of being prophylactic about it, do some sort of early detection and then spot treat, and then simply survey and treat along as you need to.
Yeah, and that's the position of the Sustainable Beekeepers Guild, is that, regardless of the treatment philosophy, wherever you end up on that spectrum from one end to the other, that you are monitoring and evaluating whether or not you should be intervening in a specific way, not just intervening for the sake of treatment. Regardless, you got to have some information or some data before you act.
Yeah, and again, if I have 100+ colonies, sampling is, you still have to do it, but it's really hard to do it on every colony, right? It's when you have 10 or less, and especially when you're in one yard, you really should be doing that. You can get that much more information out of your yards and probably take care of your problems much quicker. And the next question is, well, with this new information, should we reimagine what alcohol wash data means at different times of the year? And I think Jack Cameron really has a good flow chart that he does for some of the extension work, where I'm pretty positive it's Jack Cameron, where he has it, if you're getting a detection early in the season in an alcohol wash, that's probably going to be really high later on. And he recommends a different management strategy than if you just find a detection 3/4 of the way through the season. And you might be able to modify some of that information based upon your location and get some better results than what you're getting now. I don't think we necessarily have to wait for that 3% economic threshold.
So when you're testing your drones with the eye test, or the monitoring method that you're hoping to kind of improve upon or systematize, when you're doing this, are you just doing the adult drones, or also looking at drone larvae?
We're just doing adult drones, and with the citizen science project that James Wilson and I are doing in Virginia, we're asking the members to essentially take 2 frames of brood and just take 40 drones between those 2 frames. So essentially 20 drones per frame. Not all colonies are going to have that many drones, by the way. They're not always emerging, and we only want those young ones in the center of the brood nest. Just take 40 of them and throw them into alcohol. Just have an open deli container of isopropyl. Everyone says ethanol. Just get some isopropyl. It's cheaper and easier to get. And the drones will die in it like that. And all you have to do is shake it for about 30 seconds after you throw 40 drones in it, and the mites will come right off. So you can get the detections really quick. And every time I survey people, how bad do you feel about killing 40 drones? And people are like, I'll do it again. How bad do you feel about soaking 30 nurse bee? Oh yeah, exactly. And
four drones.
Yeah. And it's the funny thing about us. Hey, I see some good questions here. I want to jump on them.
Yeah, go for it.
Is it pheromone that draws Varroa to adult drones? Scott, I brought in 3 grants to look at and answer that question. And it's a huge thing that we're looking at. And I proposed it really early on. Essentially, we want to see if we can come up with a bait trap for Varroa. That's our golden goose, our golden ticket, right? Especially for commercial beekeepers. If I can hand them sticky pads, they toss it in their hives and the mites are attracted to them. So we are really interested in the cuticular hydrocarbons of drones, cuticular hydrocarbons of mites, or the aromatics they might be releasing, to see if we can make a bait trap. So we did work all last year looking at aromatics, and now we're going into cuticular hydrocarbons of those hosts, and we want to see, can we identify them? How unique are they? Can we synthesize them and make a trap? And these are far more difficult things than they are to do them to talk about, but we're in the process of it. And it's a really good question. What sample size do you hand count for drones? 40 is great. And not all colonies are going to have up to 40.
So that's 40 drones that you're sampling, but then how many mites are you looking for? A particular number of mites before you're like, oh yeah, I'm tossing this.
Yeah. So if you're not a member of ABJ, become a member of ABJ. It's a great magazine, but I have an article coming out in May on this topic. We're essentially doing it ahead as a non-peer reviewed press release because it's so important for beekeepers. And we talk about it right in there in that article, which is we don't have established thresholds yet. We don't know what 2 mites on 40 drones means, not yet. And it's likely to be very different based upon your geographic location. So it might be very different for someone in coastal Georgia compared to Wisconsin or Michigan or, or California. And so you might have to do the due diligence on your own, or what we're hoping for is there will be interest in bee labs across the country. And each bee lab is starting to do work on behalf of beekeepers and stakeholders to come up with treatment thresholds or predictive thresholds. Right now, we know if you have multiple detections, so what that means is I get, let's say out of 40 drones, 2 of them or 3 of them have mites, it's going to be infested come the fall. So we're there right now, but any better specificity, we don't know yet. I will also point out, because this usually comes up in a question, and it probably is here somewhere, drones are the most likely bee to drift from a colony into another colony. So colonies readily accept drones. They'll kick out workers, they'll beat them up, but they'll let drones right in. We had drones as young as 3 days found in the apiary across the yard, across the shed in like another colony. They drift at really young ages too. So it's possible these colonies that were getting these detections on drones early in the season are actually seeding healthy colonies nearby with low levels of Varroa. So when I say removal could be a really healthy strategy, you might be conferring protection to all of your other colonies by simply breaking them down, treating them, requeening them, and going forward with those colonies not in your yards, essentially. Not a question, but the chat is closed. M&A's made so much sense. Thank you. Thank you, Kyle. I appreciate that.
The M&Ms was a great idea. Yeah. Piggyback on the drones and the low level mite drifting. Excuse me. If the colonies that are weakened or are showing an inability to tolerate the Varroa loads or to keep them down or show resistance or whatever host of mechanisms the bees have to handle the mite loads, how much of an impact would that have, let's say, on your local DCAs, especially like in my case, I'm flooding my DCAs, at least in theory, with a lot of my own colonies. So what kind of impact would that have on, improving my open breeding approach if I'm culling out all these drones all the time?
Yeah, so I would look at it this way. You can do repeated alcohol washes or the drone sampling and identify colonies that don't have mites in the drone population, right? So if you really need a lot of drones, don't cull those drones. But if you have colonies that you're getting regular detections of mites on the drones, you should absolutely be culling those drones. One, because you don't want them in your breeding pool anyways, right? You don't want a potential vector of viruses directly to your queens through vertical transmission. And you're simply allowing Varroa to reproduce. So if you have a small number of colonies, so if you're, 10, 20, whatever, you could totally do this if you're investing into your own queens. The problem is if you get into a much larger number, if you're producing thousands of queens and you've got hundreds of drone colonies, it's much more difficult to do something like that. But I don't think you're going to run into a problem with a slightly nuanced management to have tons of drones. And that's never come up. And I used to do drone comb removal religiously in my colonies. Claire asked, what happened to that colony that had the 60% parasite burden? Every single colony, every one of those 5 colonies, they all died. So 60% was the highest that got parasitized in 9 days. Even the ones that were lower at 18%, they all died. So every single one of them passed away. And every single one of them was treated at the end of the study. So we didn't just let them ride till the winter. We treated them and we fed them. They still died. And they looked like a normal collapsed colony. You eventually go in, it's smaller, and then you go in and nothing's there.
If the alcohol washes with worker bees are not going to really give a good accuracy to the early season monitoring, what if you had to choose, what frame of bees would you use then at that point?
What's that? One more time.
So with the inaccuracy of the alcohol washes because of the aggregate mites on drones, what frame of bees, if not nurse bees, should you choose now?
Yeah, so I don't, so to answer that, I'm just going to say, I don't think we should throw the baby out with the bathwater. Right now, we know, we have some relative idea of how many mites on an alcohol wash mean, right? So keep doing it. What I'm saying though is it's going to be inaccurate at some level because it's designed for one type of distribution when we actually have a different type of distribution in the colony. And Calderon already showed this. He did repeated alcohol washes on the same colonies. And he had wildly different results, even though he's just grabbing different bees from within the same colony. We don't know that answer yet. I don't think collectively as a research group do we have that answer. And the problem is he had, I think it's 200% above and below a mean he would get from these repeated alcohol washes, which theoretically means you could have a 1 and not be worried about the colony, or you could do a repeated alcohol wash, have a 3 or 4, and actually you should be worried. So, I don't really know where it goes. What we're essentially bringing up right now is here's a problem. We can't ignore it. That's what that comes down to. And we really point this out because especially commercial beekeepers, they're getting hit on all fronts right now. They're dealing with heat stresses when they move their bees. They're dealing with lots of pesticide exposure when they're paid to do these pollination contracts. They're repeatedly managing for their mites and treating. And they're not just throwing chemicals in, walking away. These are beekeepers that are regularly testing. They're rotating their treatments. So they're not just relying upon a single treatment, they're rotating them. They're using multiple treatments to cover one brood cycle. So they're getting all the Varroa and they're still getting bad results. So we really want everyone to be aware of the issue and not ignore it in any way. Because unfortunately for a lot of these beekeepers, it's going to continue to be more costly before it gets better. What I can say is as hobbyists, we have that ability with lower numbers to be more nuanced in our management. So.
Yeah, that's a lot of food for thought. So we should still be extending some grace to the current methods we're using to determine whether or not we should intervene, especially as far as the mite wash is concerned.
Yeah, let me answer these two questions at the same time. So Michael Lund and Karen Eaton. Michael Lund brought up our club had mite count go from 1 mite, 8/14/22, to 35 mites within a 1 month period. So between August and September. Is this possible? Low mites for the previous 16 month, our dearth is July. And then Karen said, so the mite bomb from outside colonies is now debunked. The mite bomb is within the colony, right? Okay, so let's look at the first one, which is, well, those were your results, so it's totally possible. It could be possible because of that was just the natural mite growth, or it could be possible from a combination of factors like your drones disappeared and now there's a shift into distribution. You're simply collecting more bees that have mites on them. Within that period of time, you also had natural mite growth. You also could have had emigration of mites into that colony during that period of time. Okay, all of those things would increase the number of mites in the colony or the number of detections. You also could have had something happen like, let's say you just lost tons of foragers for some reason. They just died off, pesticide exposure. They got hit because a parade went by your colony on a day where there was a flow. I don't know. Those foragers are really unlikely to have lots of mites on them, right? But all the bees in the colony are more likely to have mites on them. So you would have a shift in the proportion of mites to workers or to host. What I'm saying is there's a lot of confounders that over a period of time could actually skew or alter your alcohol wash. But what you saw was this giant increase. Now I'm sorry, it moved on me. Was her name Karen Eaton? Brought up this debunks it doesn't debunk it. We know from several research projects that mites are absolutely moving from one colony to another. And I would argue that mites have to engage in dispersal, in inter-colony dispersal to survive. Seeley showed that instead of mite bombs, it's really more like robbing lures. And so think about what he showed. They had colonies that were robbing out other collapsing colonies, bringing the mites back to their colony, okay? And so if we were to think of it from a mite's perspective, look, my house is on fire, it's collapsing on me, and these healthy smelling bees enter the colony. They simply jump on them and return to the other colony. You would get a spike in your alcohol wash level from that. At the same time, Kelly Kilhannock did show that collapsing colonies will send out bees into other colonies which are accepted. And there totally can be a low level of mites on those older bees. So it doesn't debunk it, but what we show is a more parsimonious answer for why a lot of your colonies would have a spike in alcohol washes come fall. And I think we really want to focus on that parsimony, where it's the easier to explain answer for what's happening within the colony, but we shouldn't exclude these environmental factors that are also causing emigration into these colonies.
So we're not completely letting the mite bombers off the hook, but at the same time, we can't necessarily blame all of the mite bombers for the mite increase in the end of the season, because that increased number is not just all of them coming out from the cappings, it's that they've emigrated from the drones to the workers.
Yeah, and I don't like mite bombers because I've heard so many beekeepers blame other beekeepers for the mite bombs, and it's kind of become this way to blame someone. And really what I think it should be is use more nuanced language. We have an extremely complex host parasite pathogen system with a really complex pathogen. So let's have a nuanced conversation. We don't want something that complex to just get clickbait. We're better than that. We really are better than that as beekeepers. And I expect better from beekeepers. So instead of saying, mite bombers, say, apiaries that did not engage in proper removal of mites or proper removal of an infectious vector. If we start having conversations like this, with that little bit more detail, we could probably change human behavior. And so that's how I want to respond to it, because it's kind of been so ingrained in us to use almost clickbait language, but then we lose the nuance in the conversation that we really need to have. And so no, but it doesn't exclude the fact that emigration of mites into my colonies, your colonies, could be occurring because colonies that were not managed for an infectious disease and an invasive pest weren't removed from the population at an earlier date.
Yeah, I appreciate your comments on that. It's my personal feelings that even if somebody has a colony that degrades over time or becomes a potential robber lure, like Seeley and Peck called it, but that we do engage in that nuanced dialogue about what's happening there. Because even that person who has a colony that's gone into this status of epic failure, they have probably had good intent at some point. So it's not just an act of maliciousness. It's often attributed to that term. So I definitely appreciate that.
Yeah, and we don't want to alienate anyone, right? So we have one person that says, I'm a queen producer. I don't like wasting drones, but I'm part of the VA Citizen Science Project this year. And I would just respond to that. We're asking you to collect 40 drones per colony. Your colony, a healthy colony, Seeley showed, should have about 500 mature drones at any point during the season where they're the swarm season. You're fine. It's not going to reduce any of your mating at all in any way. And if for some reason it did, you just didn't have enough drones to begin with. So still do it. Claire brought up something really good, which is, hey, if I find 2 or 3 mites on my 40 drones, what would be the best way to treat that colony? And it's impractical to hand pick up all the drones. And so what would we treat with? Well, early in the season, April, May, and I say April down here in the Mid-Atlantic, not April up north, right? You only have a couple days to really reliably get into your colonies and take brood frames out and safely leave them out. You also might not have enough drone production at that point to get a reliable sampling method. So May is great. Well, what are your temperatures in May? They're still cool enough for formic acid. Formic acid is an excellent spring treatment, especially if you notice, I always say split, treat, and then requeen. I can take these smaller colonies and apply a treatment in a smaller area. It's probably going to become more effective. And you can either put the queen in ahead of time and treat or treat after. But that's probably a great way to go. So I think formic is excellent at that time. You probably don't have enough time for apivar. You could do repeated alcohol or oxalic drizzles. I wouldn't worry about the queen's health at that point. And the reason being is we're going to requeen her. She already proved to us she can't suppress mite reproduction. That should be part of everyone's breeding program. So treat. If the treatment ends up damaging your queen, that's okay. You're requeening it anyways. And that's the attitude I've taken. And the reason I'm suggesting this, if you have few colonies, is those heavily infested colonies are devastating to you. They're also really expensive. So you can have a $200 loss in the fall, or you can make $200 to $600 worth of splits in the spring. Which one do you want? And so what I'm doing is I'm taking my really big colonies that are getting multiple detections, turning them into, if I just do a split, I get a split. That's $200 gain. Or if I make nukes, I'll get up to 6 nukes from them. There's $1,200 worth of new colonies instead of taking the $200 loss in the fall. So the economics are really in my favor at that point once I decide to do the split and treat. So you can make a little bit of a mistake and you're still going to get paid out well.
Looks like we got a couple of more. I think this is the most questions that we've had at one of our presentations, Zach. Very well
Awesome.
done. Generating a lot of good positive thinking. Does it help to select open brood drone or worker frames and shake all the bees into a plastic dishwashing basin, allowing the foragers to fly off while nurses just walk around the walls of the basin? We get much better results selecting from primarily nurse subjects.
So I get what you're doing there, which is you're letting the likely bees that don't have mites on them to leave your possible sample so that you get a better detection. The problem is we still showed that those nurse bees are still unlikely to have mites on them compared to the young adult drones. So it doesn't answer that question. And then later in the season, when you're doing your sampling, when drones aren't available, it really wouldn't matter, right? We're getting detections largely on those first, second, third, fourth day old bees, and we get fewer on the older bees. So sure, your foragers let them fly off, which is probably happening anyways. But by that point, you're essentially just trying to find high levels. So I see what you're doing there. It's a really good thought, but it still doesn't answer the problem of early in the season that distribution's on adult drones.
Zach,
So
absolutely brilliant. Excellent work and excellent presentation.
Thanks. And so I just have open-mated carniolans. That's what I use for my research project. Do you think the bees infected with numerous viruses have a negative impact on the new AFB vaccine? I think we're looking at 2 different pathogens there, and that's for someone else to do the research on. I don't study AFB and kind of don't look at co-infestants like that. It's a good question. You're welcome. Glad to do the research. I'm considering collecting pollen this spring, but it seems like drones are confined in my hive during that period. Are drones causing lots of trouble in my hive? Are drones causing lots of trouble? Probably not. So in and of themselves, drones are an integral part of your honeybee colony, and your worker bees want them there. And I really point this out. Drones have a really, really important job, and that is to successfully mate with a queen, not from their own colony. If they do that, your colony is a huge success story. So let's think of it this way. There's 62 people still in this chat. If we're all honeybee colonies and my colony is the only one with a drone that mates with other queens, I'm more successful than all of you. Hands down. Doesn't matter how much honey you produce. It doesn't matter if you survive the winter. I could die in the winter. I'm still more successful if my drones are the successful ones. So your colonies want those drones. Are they doing anything bad? No, likely not. They're eating food and they're begging for food. And when they get older, they simply aggregate on the outside frames. Okay, that's about all they're doing. If mites are moving on them, then the mites are feeding on them. It's really not causing any harm to your worker bees. And that's about it. The only thing that I haven't addressed there is for really long extended times, do they start defecating in the hive? Mm-hmm. That's a really good question. And that could be a fecal to oral transmission route if the drones are infected. So that'd be like the one thing I'd think of. Thank you for your family and the efforts in helping with the research. Give them a big jar of honey and some mead. My mom is actually a beekeeper now, so she gives me the jars of honey, which is the irony of it. So, but anyways, everyone, thank you so much for having me on here. This was a blast, and I look forward to it. And James, thank you so much for the organization putting this on. Yeah, we're glad that you came by, and glad that we were able to catch, the summary of the research that you've been working on for so long. And I've had the privilege of sitting on some of your queen talks way back in the day with Ange Roll and some of the others over there with their Bee Breeding Alliance. And so the presentation was very helpful, and I was encouraged by
your demeanor and your approach. And so I had high expectations, and you definitely delivered. Very grateful for that. I do want to know if you're going to give us a preview of if you're making any suggestions on the mite numbers for your May article with ABJ.
One sec, Chris, I didn't know you were on here. I love you. I miss you from Oaxaca. No, we don't have any treatment threshold recommendations, and I openly acknowledge in the article that it's likely to be geographic specific, and beekeepers will likely have to do some due diligence, and we're really encouraging it. I would love to hear that a research lab in the Southwest, in the Midwest, Pacific Northwest, et cetera, Chris down in Oaxaca, come up with their own treatment thresholds because they're doing their own work. The nice thing is this isn't extremely expensive or difficult or time consuming.
So that circles back to the very beginning when we talked about being diligent and purposeful in your beekeeping efforts. And that is kind of in line with what the Sustainable Beekeepers Guild is promoting and actively engaging with our mission. And I guess we'll close with this question, Zach. The board members were texting me throughout the duration of your presentation. They would like to know if we could offer you a lifetime membership to the Sustainable Beekeepers Guild. However long the Bee Guild might be around, we're hoping a really long time, but a lifetime membership, has endless value at this point.
I will take it. That is awesome. Thank you very much.
Oh, spectacular. We're happy and humbled to have you. Thank you so much for coming tonight, everybody. Thank you so much, Zach. I'll catch up with you here soon, and we'll talk to you, everybody else, later.
Sweet. See y'all.
Have a good night.